mind. 떨리는 지금도 you're on my mind all the time. I wanna tell you but I'm super shy.
Welcome and thank you for joining us today. In this comprehensive presentation, we will embark on the journey into the intricate world of lipids and related systems. So, our objective is to provide you a deep and informative understanding about the biosynthesis, transport and shuttle systems, structures as well as their functions and classifications, also the physiological significance and its clinical correlation. So first, what is lipids? So lipids is derived from a Greek word lipus, which means fat. Biological lipids are chemically diverse group of organic compounds which are insoluble in water, soluble in non-polar solvents such as ethers, chloroforms, or benzene. Unlike the proteins, nucleic acid, and polysaccharides, lipids are not polymer. Fat and oils are principal stored forms of energy in many organisms. But first, what is biosynthesis? Biosynthesis and its transport and shuttle system. So we will tackle first what is biosynthesis. Biosynthesis is the process by which living things use chemical reactions to create products useful for cellular metabolism. On the other hand, it is the living things that makes important molecules and compounds used for survival and proper functioning. Biosynthesis reactions are also known as anabolic reactions because the final product are large, complex structure called macromolecules. So the pathway of lipid transport. There are two types of pathway of lipid transport. So first, we have the exogenous pathway. So first, the first steps is anita sa intestine na, where ang fat and cholesterol have been absorbed sa gastrointestinal tract and then mo-assemble sila to form chylomicron. And then, ang chylomicron mo-travel ni siya into bloodstream to peripheral tissue or to lipoprotein lipase to adipose tissue. And then, kaning chylomicron mo-release rin siya fats once na-meet niya ang LPL or lipoprotein lipase. Also, in these steps, these allow fats to absorb in the form of fatty acid and glycerol. After unloading their fats, kanidang chylomicrons will become siya smaller and then known as chylomicroruminants. And then, kanya siya anti-HDL is paano ni siya no? Byproduct ni siya sa step 4 and step 3. So, we will proceed chylomicroruminants and then travel ni siya to the liver. But, because of the virtue ApoE, mubind siya sa ruminant receptor, maunang mo-liberate siya sa either cholesterol or free fatty acids. So, that is the process sa exogenous pathway. So, we have the second one, the endogenous pathway. So, sa endogenous pathway, so, once naabot na ang free fatty acid and cholesterol sa liver, more repackage ni siya to form from VLDL or very low density lipoproteins. So, kaning very low lipoproteins, mo-travel ni siya into lipoprotein lipids and then mo-adipose siya og tissue. But ano nga time sa exogenous pathway, mo-absorb ba siya no, di ba? Mo-absorb siya og fatty acid and glycerol. Sa ani nga time, mo-release siya no, iyahang i-release ang fatty acid and glycerols. And then, once ang fats is mo smaller siya, mahimo siya, ang tawag siya og IDL or intermediate density lipoprotein. And then, so kaning anti-HDL is byproduct lang gihapon ni siya. So, IDL, mo-absorb siya sa blood from the liver and then mudong siya sa hepatic lipase. So, ana siya ma-break down and then mahimo siyang LDL, meaning stands for low density lipoproteins. And then, mudong na siya mo-absorb ang LDL of various tissue with the help of LDL receptor. And kaning LDL mo-absorb na siya sa liver with the help also again with LDL receptors. So that's the process of exogenous pathway to endogenous pathway. And we will now tackle about the shuttle mechanism or system. So the shuttles are the system used to transport the reducing agents across the inner mitochondrial membrane, and it helps in the transfer of electrons from NADH to electrons transport chain. Then, by the way, so ang NADH is made in glycolysis, which happens in cytosol and cannot across the mitochondrial membrane. The reduced electron potential energy must get into mitochondria in order to deoxidize in the electron transfer chain to make ATP. So we have two types of shuttle. So, shuttle mechanism. So the first one is the malate aspartate shuttle. So it's simply called malate shuttle. Part of the citric acid responsible for energy metabolism, mainly used to transport the electrons produced during glycolysis to inner mitochondrial membrane for oxidative phosphorylation. Provides an important mechanism to regulate glycolysis and lactate metabolism in the heart by transferring reducing equivalents from cytosol into mitochondria. And also, the malate aspartate shuttles is mo-operate ni siya sa ato ang heart, sa atong liver, and kidney cells. So, we have the process of malate aspartate shuttle. So, we will begin with oxaloacetate, then mo-oxidize ni siya to malate with the help of cytosolic malate dehydrogenase. And then, from NADH+, mo-reduce ni siya to NAD+. And then, mo-travel ni siya into mitochondrial matrix. And then, sa aning nga time is, mo-reduce na siya og equivalents. So, it can lead to oxaloacetate with the help of mitochondrial malate dehydrogenase. So, from NAD+, mo-produce siya og NADH+, H+. So, sa oxaloacetate, mo-undergo ni siya into transformation reactions. From glutamate, mo-convert ni siya to aspartate and then to alpha-ketoglutate. And then, mo-travel ni siya into intermembrane space and then, balik lang gihapon siya, mo-undergo gihapon siya og transformative reactions. So that is the process of malate aspartate shuttle. We have the second one, the glycerol 3-phosphate shuttle. So it is a mechanism that originates NAD+ from NADH, a by-produced product of glycolysis. The shuttle consists of sequential activity of two proteins. We have the GPD1, which transfer an electrons pair from NADH to dihydroxyacetone phosphate or DHAP, forming glycerol 3-phosphate and originates NAD+ needed to generate energy through via glycolysis. And also, the glycerol 3-phosphate shuttle was first characterized as a major route of mitochondrial hydride transport in the flight muscle of blowflies. Plays also an essential physiological role in yeast, plants and animals, and also it's so the vital participants in biochemical reactions. So these are the process of glycerol 3-phosphate shuttle. So from dihydroxyacetone phosphate mo oxidize siya to glycerol phosphate from NADH with the help of NADH plus H plus to NADH, and also with the help of glycerol phosphate dehydrogenate. And then, undergo to siya to mitochondria and to balik siya dihydroacetone phosphate with the help of FAD to FAD2 and also by glycerol with the enzyme of glycerol phosphate dehydrogenase, and mubalik lang gihapon siya. So we are now in lipid structure. Lipid structure, fats, oil, waxes. Yes, it provides energy for cells, cell structure, insulation, and lipids and proteins compose the cell membrane. So cholesterol gives cell membrane flexibility. So sa structure sa lipid, naa siya duha ka part. The head is the glycerol, and these three is the fatty acid molecules. The monomer is the fatty acid, and polymer is the lipid. We are now in lipid functions. So, lipids are diverse group of molecules that serve various essential functions in living organisms. So here are some key functions of lipids. So energy storage. Energy storage in organisms in the form of fatty acids. And we have structural components. It provides barrier between the cell's interior and its environment. And so we have hormone production, helps and regulate growth, development, reproduction, and metabolic process in organism, and absorption and transportation from or of the cholesterol and free fatty acids, and buoyancy and flotation, muhatag siya og balance and flotations. These are just few many important functions of lipids, but overall, lipids are essential for proper functioning of cells, tissues, organs, and the overall health and well-being of organisms. So, classification of lipids. So, lipids can be classified in variety ways. In one categorization, they can be divided into two major classes. We have saponifiable and non-saponifiable lipids. So, non-saponifiable lipids, they are lipids that cannot be broken down in the smaller molecules by hydrolysis. And this includes the waxes, triglycerides, phospholipids, and phingolipids. And we have the second one, the saponifiable lipids. These are lipids that have one or more ester groups, enabling the process of hydrolysis to take place but with the help of substance like acid, base, or enzyme. So lipids is classified in four classes. We have simple lipids, compound lipids, derived lipids, and miscellaneous lipids. So first, we have simple lipids that made of fatty acids and plus alcohol. Example, this one is the tag and waxes. Tag is the neutral fats, and waxes is gamit yun kay siya sa pharmaceutical industries like lotions, ointments. And we have compound lipids, fatty acid plus alcohol plus additional group. Example is the phospholipids, glycolipids, and lipoproteins. Lipoproteins is lipid plus proteins. So we will tackle this one later on. And we have the third one, derived lipids, obtained by hydrolysis of simple compound lipids. Example of this one is the fatty acids, steroid, eicosanoids, and ketone bodies. Eicosanoids is kadto ni siyang mga prostaglandins, leukotrienes. And we have the last one, the miscellaneous lipids. Possess characteristics of lipids. Example is the squalene and carotenoids. So we have fatty acids. Fatty acid is the simplest form of lipids. These are monocarboxylic, straight, unbranched hydrocarbon chains contain even number of carbon atoms between 4 to 36. So fatty acids are also known as acyl group when it is part of esters. So fatty acids are amphipathic in nature. And the fatty acids may be free or esterified with glycerol from triglycerides. So we have two types of fatty acids. We have saturated fatty acids and unsaturated fatty acids. So example of saturated fatty acid is atong mga dietary products, mga meat, butter, and unsaturated fatty acids ay kadtong mga vegetable oils. So, the difference of these two. So, what are the difference of these two? So, ang unsaturated fatty acid is wala siya double bond. But the unsaturated fatty acid is naa siya double bond. And this one is solid in room temperature, while unsaturated fatty acid is liquid in room temperature. Also, saturated fatty acid has more hydrogen than unsaturated fatty acid. As you can see, saturated fatty acid has 18 hydrogen, while unsaturated fatty acid has only 16 hydrogen. So, essential fatty acids or FA, fatty acids that humans and other animals must ingest because the body requires them for good health but cannot synthesize them. Deficiency can cause dermatitis, membrane function loss. Arachidonic acid is essential when linoleic acid is deficient in the diet. So we have two types, the linoleic acid or omega-6 essential fatty acid and alpha-linolenic acid or the omega-3 essential fatty acid. So first, we have linoleic acid or omega-6 fatty acid. Long-chain polyunsaturated fatty acid with double bond starting with 6 carbon from methyl end. They reduce also serum cholesterol, and the major source is the vegetables and nuts. So we have the alpha-linolenic acid or omega-3 fatty acids, which are essential fats that beneficial for health, and they are found in foods like fish, flaxseed, and walnuts, and are known for their role in supporting heart and brain health. It is also believed to have beneficial effects in a number of chronic disease, including cardiovascular disease, rheumatoid arthritis, and dementia. So, triglycerol or TGS, also called fats. 3 fatty acids are bonded to glycerol molecule, constitute majority of dietary lipids, stored in adipocytes or fat cells, as energy reservoir and not component of cell membrane. Subcutaneous layers of fats provide thermal insulation. So we have steroids. Steroid is derived lipids. Complex molecule consists of 4 fused carbon ring or 17C, phenanthrene nucleus, 3 6 carbon ring, A, B, and C. Then cyclopentane, the D ring. Then we have the 10 and 3 methyl, and the 17 is the side chain. And common cyclic ring called also a steroid nucleus. So we have glycolipids. Glycolipids are also known as galactosyl or glycosylceramide. It is contained of fatty acids, sphingosine, and carbohydrate, widely distributed in every tissue, particularly in brain. Example is the cerebrocytes and gangliocytes. So in the biochemical component of biological membrane, cell surface receptor, glycocalyx, and cellular communication, cell-to-cell recognition, and receptor for hormone and bacterial toxins. Example is the cholera toxins. So we have cholesterol. So cholesterol is the major sterol in animal tissues. Body cholesterol contains of 50% synthesized in body, 700 mg, and 50% from diet. 70 kg man is naa 140 mg cholesterol. So, no vegetable oils contains any cholesterol. So, example of mga naay cholesterol is, the common one is ang egg. So, mga egg, mga meat, yes, fatty meat, and so we have the phospholipid. Phospholipid has two classes. The first one is the glycerol phospholipids, which contain glycerol backbone. And we have sphingophospholipid na contain siya of sphingosine. And glycerol 3-PO is bonded to two fatty acid chain. The PO4 group is linked to a hydrophilic group. And also phospholipid is amphiphilic in nature, hydrophilic tail, and hydrophilic phosphoryl heads. Transport of plasma membrane. Plasma lipids are transported as lipoprotein particles. Lipids plus protein. And protein part is the apolipoproteins or apolipoproteins. Examples is the apolipoprotein ABC. Functions is lipid transport, enzymatic functions, and ligands for receptors. Lipid part contains siya of lipid of various type. And then we have the types and function of lipoproteins. Lipoproteins is chylomicrons and transport is DHEAS, and very low density lipoprotein or VLDL and endogenous TGS, and we have LDL, the low density lipoprotein, and free cholesterol sa yung transport, and ang lipoprotein is high density lipoprotein and cholesterol esters. We have the last one, clinical correlation. The drug orlistat for weight loss inhibits pancreatic lipase, leading reduced TAG, malabsorption, and weight loss. Malabsorption of lipids that are steatorureum, and then yung result is from bile acid insufficiency. Example is the liver disease, gastrointestinal retention. While in the pancreatic insufficiency, we have pancreatitis. So, and that would be all. I hope you guys have learned something about my report today.